All right, guys, so today we are going to look at the molecular basis of inheritance, which is DNA. We're going to look at how it was discovered and how it is replicated. So it was first discovered with T.H. Morgan's group. They determined that two components of a chromosome are DNA and protein, and they both became candidates for that genetic material that we pass on from parents to offspring. And then Frederick Griffin did an experiment using pathogenic and non-pathogenic bacteria back in 1928 and showed that even a killed pathogenic, the living S, that kills mice when it's not heat killed, when you put it with the non-pathogenic R cells, that they could transfer DNA between each other, and that one could become pathogenic again and kill the mouse again. So he helped to solidify the evidence that DNA was our genetic material. And then later, work by Oswald Avery and others also identified DNA as a transforming substance. But it was really solidified with the Alfred Hershey and Martha Chase experiments in 1952 using T2 phage, which is this. And the T2 phage is a virus that infects bacteria. And there's a nice animation on the T2 phage and on the Hershey-Chase experiments within your study area of Mastery Biology. And in your text, here is a diagram showing how the experiment was carried out. But at the end, what they said is that they concluded that the injected DNA of the phage provides the genetic material. So this solidified the fact that DNA was our hereditary material. And then the gentlemen, Watson and Crick, James Watson and Francis Crick, who eventually got the Nobel Peace Prize for really solidifying, showing the shape of DNA, introducing the double helical model, and confirming the hereditary information directs the development of our biochemical, anatomical, physiological traits, showing that it codes for proteins, is all the work they did starting in 1953. And they were strongly reliant in the structure of the double helix on the work of Rosalind Franklin. She did this X-ray crystallography, this diffraction photograph, that helped to show the shape, that it was a double helix. So Watson and Crick first to determine the structure, but Maurice Wilkins and Rosalind Franklin were the first to actually photograph it, or X-ray crystallography it. And they used Franklin's work to, Watson used it to deduce that DNA was a helical, also used it to deduce the width of the helix and the spacing of our nitrogenous bases. And so that work helped to determine which of our nitrogenous bases, or which of our nucleotides, matched up with who. And so we have our 4 nucleotides. They are all identical in the fact that they have a phosphate group, the deoxyribose sugar, and then the way they differ is in this nitrogenous base. So we have thymine, guanine, cytosine, and adenine. Now remember that in RNA, the adenine is replaced by uracil, or thymine is replaced by uracil, but it matches up, the U matches up with the adenine in RNA. And here's that double helical structure. But with that X-ray diffraction, it showed how wide the helix was. And so Watson used that to determine that 2 purines couldn't go together because they're going to be too wide. 2 pyrimidines couldn't go together because that'd be too narrow. But if you put a purine and a pyrimidine together, that was what would match up. And you can see the purine is the 2 circles, the 2, I can't think of the word at the moment. But so these are our purines with our 2 ring structures. That's what I was looking for, 2 ring structures. And our pyrimidines with the single structure. And so the pyrimidine thymine hooks up with the purine adenine, and the purine guanine hooks up with the pyrimidine cytosine. And that goes back to Chargaff's rule also. And so with that X-ray data from Rosalind Franklin, Watson and Crick were able to determine that pyridines and pyrimidines came together. Watson was also able to show that the DNA was anti-parallel, which means that one strand runs 5' to 3', and the other strand runs 5' to 3' in the opposite direction. So you have a 5' end on this side to a 3', and then the 5'. And your book mentions it, it's not shown on here, but your book says think of it as a 2-lane divided highway. So traffic on one side runs this way, traffic on the other side runs that way. And so that's what the anti-parallel is that the paired strands of DNA, or the paired single strands of DNA, match up, but they run in an anti-parallel direction, so they run in opposite directions. And the bonds that hold these together, so we have our covalent sugar phosphate bond that holds the individual nucleotides in their string structure, and so they hold the phosphate to the corresponding hydroxyl group of the next one. So that's a covalent bond. And then we have hydrogen bonding that holds the matching nucleotides together, and also between nucleotides as they run down the string, we have these van der Waals interactions. So lots of different bonding holding the DNA in that double helical structure and allowing it to condense and exist as it should. So here again, back in 1950, Erwin Chargaff proposed 2 things. He had 2 findings. He said that the base composition of DNA varies between species, but that it is all made up of the same nucleotides, and that any species, the percentages of A and T were equal, and the percentages of G and C were equal. And that is solidified when Watson-Crick showed that A and T match up and G and C match up. And so here is the pairing of the nucleotides. And so also with the research into DNA, Watson and Crick were able to prove, and later experiments by Matthew Meselson and Franklin Stahl, Meslin and Stahl, supported the semi-conservative model. And so what the semi-conservative model is that each strand of DNA, when it is taken apart, is replicated, and then each new daughter strand is made up of one old strand and one new strand. And this continues on, and every new replication has one old strand and one new strand. So take the original, open it up, replicate it, and then the 2 new daughter strands have one old strand, one new strand. Other things that were tested were the conservative model, where the 2 old came back together and 2 new went together, and a dispersive model where it mixed and matched. But the one that was proven was a semi-conservative model, and this also is essential to the very low error rate that we see in DNA replication, because you have a proofreading built in. You're matching the new strand to the old strand, which automatically proofreads the new strand's replication. So how is DNA replicated? So we're going to look at a comparison between E. coli, which is our prokaryotic cells, and a eukaryotic replication. Remember that prokaryotic organisms have circular DNA, and that eukaryotic organisms have linear DNA. And they both start with an origin of replication, but since the prokaryotic DNA is a much more simplistic circular piece of DNA, you just get one origin of replication. Origin of replication starts, opens up the double helix, and then starts to lay down new nucleotides, working its way around the circle until you get 2 new daughter molecules. With eukaryotic, because it is linear and a lot larger than the prokaryotic, you actually have multiple origins of replication. And this area that's opened up is called our replication fork. And so the DNA replication moves in both directions away from that origin of replication until all of them meet up, forming two new daughter strands of our DNA molecules. So this is with our linear chromosomes. And so some of the primary enzymes that are involved are, first we have to unzip. And so when we unzip the DNA, we are using an enzyme called helicase. So helicase unwinds and separates the parallel DNA strands. Then it lays down some single-stranded binding proteins to stabilize and keep the strands separated, because we are untwisting that double helix to help break and stabilize and keep from supercoiling the DNA that is not being unwound. We use an enzyme called topoisomerase. You also sometimes see it called gyrase. And so this helps to keep the DNA that is not being separated from getting too supercoiled, too kinky and curled as you unwind the rest of the DNA. Another essential enzyme is primase, because you're going to see that the enzyme DNA polymerase that lays down the new nucleotides, so DNA polymerase 3 will be the one that does that, needs a double-stranded area of DNA, or double-stranded area of nucleic acid to lay down, because this isn't all DNA. Notice the primer is an RNA primer. So the primase lays down RNA primers to tell DNA polymerase where to start replication. And so as replication goes, that 5' to 3' is important, because all of our incoming nucleotides are going to be added on to the 3' end. So our new DNA strand grows in the 5' to 3' direction. And that polymerase, DNA polymerase, is going to add on those nucleotides to the 3' end of the growing strand. And so DNA will only replicate, DNA polymerase will only run in the 5' to 3' direction. So you're going to see why this is important in just a minute. So because our DNA is anti-parallel, and so we have one strand running 5' to 3', it means that the new strand will grow in the 5' to 3' direction, because it's going to grow in the anti-parallel direction. And the same thing with this one, it's running 3' to 5', which means that the DNA polymerase is adding on to the new strand's 3' end. So it is running, DNA polymerase adds on to the new strand's 3' end. And so we have two different strands. We have one that can follow easily behind the helicase, because remember, helicase is unwinding, laying down the single-stranded binding proteins. And then RNA primase, the primase comes in, lays down the little red guys. These are our primers. And then the DNA polymerase 3 can bind to that primer and start adding on nucleotides. And it adds on nucleotides to the 3' end of the growing strand. And on this side is called our leading strand, or our continuous strand. And so it can just ride along behind helicase, adding on the new nucleotides as the DNA is unzipped. With the other strand, because it has to run in the opposite direction, it can't just follow along behind, because it has to add, starting here, go this way, starting here, go this way, starting here, go this way. And so each of those little red dots that you see is a primer that had to be laid down. And so with this strand, this is our lagging strand, and it is going to lay down DNA in fragments called Okazaki fragments. And so same steps. Helicase is open, single-stranded binding proteins are holding it open. The primase comes in and lays down the RNA primer. DNA polymerase 3 comes in and lays down nucleotides to the 3' end of the growing strand until it hits the next primer. And then it's going to release, go to the next primer, lay down more nucleotides. And in the meantime, you have to remove all those RNA primers, because you can't have a replicated DNA strand that's part DNA and part RNA. So we have another DNA polymerase, which is DNA polymerase 1, that comes in and replaces the RNA primers with DNA. And then once it does that, it can't make that final covalent bond, because remember, the phosphate and the hydrogens that are added together are going to be that covalent bond. And so we have another enzyme, ligase, that comes in and seals that final bond between the growing strands, between the Okazaki fragments. So leading strand made continuously, falling behind the Okazaki fragment. Lagging strand has to be made in little snippets, because you have to add on. DNA polymerase 3 can only add on new nucleotides to the 3' end of the growing strand. And so the whole set of enzymes that we use helicase to open up and unwind the DNA. We have topoisomerase out here preventing the still joined DNA from coiling and twisting. Once we open up with helicase, we lay down the single-stranded binding proteins to stabilize and keep it open. Then primase comes in and puts down our primers. Once primase comes in and puts down our primers, then DNA polymerase 3 can come and bind and grow the strand. So this is probably a better image. So on each strand, DNA polymerase comes in, elongates on the 3' end of the new strand. With the lagging strand, it has to come in and do these short Okazaki fragments. And it does each fragment. And then once each fragment is completed, then DNA polymerase 1 comes in, removes the RNA primer, adds in new DNA nucleotides, and then ligase comes in and seals that final covalent bond to make the continuous strand a single strand. So that is our DNA replication. If you have questions, be sure to talk to your instructor. And then we need to finish out with our mutations real quick. But we will stop here, and I'll do a short talk to finish that up. Everybody have a wonderful day, and look for the second talk on DNA mutations, and I will run that BioFlix video also to review over DNA replication in our next video. Have a great day.